I want to welcome everyone to the second community lecture series tonight. We have Dr. Nate Wetzel with us tonight from the chemistry department.
Bowling.
And, pardon me?
Bowling.
Bowling. Did I say Wetzel?
You did.
You know why? I'm getting these Wetzels and Mentzels and all these other German names all screwed up. This is Nate Bowling, folks. And that's on film, by the way, so I can wave it. Yes, absolutely. So, the next lecture will be on November 12th, and that's Vera Klakovkina out of foreign languages, and she's going to be talking about Starbucks, iPads, and French literature, so that should be a lot of fun. Tonight we have with us Nate Bowling. Nate has been with us for, I believe, 5 years. 2006. 2006 he came here. He is an associate professor now, and he's tenured. There's a beeping noise. And where is that coming from? Is that the door? Anyway, I'm going to read a little bio about Nate and then tell you a few little tidbits about him. Nate lived in Washington, Indiana. I know where that is, by the way, until 1996, where he graduated from Washington Catholic High School. After 4 years of study at Valparaiso University, which is pretending to be a good basketball school, right? He graduated with a BS in chemistry in 2000. From there, he continued his education at the University of Wisconsin-Madison, performing research under the supervision of Professor Robert McMahon in 2005. Nate defended his PhD thesis entitled Carbines with Multiple Triple Bonds, Synthetic Design and Spectroscopic Characterization. And the chemists in the audience are likely getting very excited by this point. I will be stumbling over a few more words here in a minute. After 2005, he worked with Professor Jeffrey Moore, and that was at Illinois Champaign-Urbana, and studying alkyne, this is where I begin to stumble, alkyne metathesis, phenylene, ethylene, macrocyte formation, and photoresponse colloidal systems. Should I repeat that? Did everyone get that? I think I said most of them correctly, but having said that, chemistry is always filled with these very interesting combinatorial kinds of words that really are kind of neat. In 2006, he joined the chemistry department at Stevens Point as an assistant professor. He's currently associate professor with research which involves designing novel conjugated organic molecules. Those are organic molecules that have alternating double bonds. See, I remember from my organic chemistry. That could replace precious metals as conductive and semiconductive elements in modern electronics. Nate teaches courses in organic chemistry and advanced organic chemistry at the college. He recently received a $250,000 grant from the NSF, and you can tell folks what that was about because I still can't quite figure it out. It has to do with polymerization and bulk properties of materials and electronic ways that molecules interact with each other. I think that was pretty cool. Yeah, not bad. I'll collect my beer later on that one, right? I want to welcome Nate Bowling to the Community Lecture Series, and we ask you to please give Nate a hand.
Thanks for the introduction, Chris. I'm a synthetic organic chemist, and what that means is when I go into the lab or when my students go into lab, what we do is we make new things, specifically new molecules. And one of the things my mom asked me when I told her this is what I do is, well, if they've never been made before, how do you know how to make them? Well, this is what I spent too many years in school learning to do, is make new molecules. And it's not really that difficult once you've learned it and you've got your PhD in the field. The really important question is, what molecules do we make? If you have this toolbox where you know how to make new molecules, things that have never been made before, you still have to figure out what you're going to make with those tools. Now, what we don't do in the lab, and this I think might be surprising to some people, is when we say we do an experiment, is I never go into a lab and mix chemicals and say, let's see what happens. Okay, that's not how we create new things. We know exactly what we're targeting. When my students run a reaction, we know exactly what molecule should be formed, and if it doesn't work, we have an idea of why it didn't work. So we're never just mixing things and let's see what happened. It's very directed synthesis. But again, we go back to that question of what should we make if we know how to make things? And as any good scholar would do, what we do is we steal other people's ideas, specifically nature. Okay, so nature has all these great ideas for things that could be made. And there's lots of different ways we can use nature as inspiration. We can use those molecules directly. We can take something that nature does and we can tinker with a little bit. We can replicate natural molecules. I'll talk about that in a little bit. And what I'm primarily going to be talking about tonight is we can just be inspired from natural molecules. Take an idea that nature has, try to replicate that process without replicating exactly what it is. So first off, let's start with using natural molecules. One of the early reports, if you want to recall a report of using a natural molecule, was Hippocrates reported that from the extracts of the bark of a willow tree, you could make a concoction that would alleviate aches and pains. And what chemists later figured out was the active ingredient in this was salicylic acid. And if you don't know chemistry, you don't need to understand what this structure means. That's just the structure of salicylic acid, so it's not a big deal. This process for isolating molecules and using them as medicines is rarely, if ever, used. So any of the medicines you take are unlikely that they come from a plant, that we extracted from the plant and they give it to you directly. Most of the things you take as medicines are made in the laboratory. Now, one problem with natural medicines is a lot of times they aren't perfect. Salicylic acid, for example, which alleviates aches and pains, can cause severe damage to stomach linings and other parts of your esophagus and things like that. So when people discovered this, what they did, when chemists discovered it, is they altered the structure just a little bit. So I call this tinkering with the natural product. Put a group on there so this molecule doesn't have those same negative drawbacks, but it still alleviates the aches and pains. It still serves the same function. And actually the active ingredient when aspirin breaks down in your body is the salicylic acid, but the aspirin is the synthetic version of salicylic acid that makes it so you can take it without eating up your stomach lining and things like that. Now, despite the fact that we almost never use natural molecules directly in medicine, there's huge effort in pharmaceutical companies to discover new molecules in nature. And what they're doing is they want to do the same thing I was talking about before, is they want to steal nature's ideas. They want to find molecules that are interesting so they can take those and reproduce those molecules. So what they'll do is, a lot of pharmaceutical companies, is they'll look for some rare species of sponge in the Indian Ocean or some plant that only exists in Borneo, and they'll extract chemicals from that, just tiny amounts of chemicals from those plants or those species of sponge, and they'll try to make those things in the laboratory once they find that those molecules are useful. And when they try to make those, what we refer to that as is natural product synthesis. So that means exactly what it sounds like. It's a product that exists in nature, and we're trying to synthesize that in the laboratory. And then a step farther, once they've synthesized those things in the laboratory, is they try to make them better. So then we tinker with them, like I talked about with salicylic acid and aspirin. If you alter the structure just a little bit, you can make those medicines better. And again, another chemical structure, don't worry about what it is. This is vancomycin. Okay? If you have a staph infection that can't be treated with normal antibiotics, you're likely going to receive vancomycin. So vancomycin is referred to as the antibiotic of last resort if you have an infection that's antibiotic resistant. Now, vancomycin fits that description I was talking about before. The way vancomycin was discovered is a scientist at Eli Lilly was consulting with a missionary in Borneo who was collecting soil samples specifically for this purpose. Collecting soil samples and sending them back to Eli Lilly so Eli Lilly could look at these molecules in the soil and see if any of them had biological activity. And what they found was this molecule vancomycin in the soil, which is produced by bacteria in the soil, could be used as an antibiotic. Now, a problem with any antibiotic is bacteria can eventually develop resistance to these antibiotics. Okay, so what happens when that happens? That's redundant. But what happens is chemists go into the lab and they alter the structure. They tinker with the structure of this. So right now, chemists are in a laboratory somewhere trying to make something that looks a lot like vancomycin, but it's just enough different that the bacteria won't recognize it. Bacteria that are resistant to vancomycin won't recognize it, and it can still be used as an antibiotic. Now, this is getting a little off topic, but just to scare you a little bit so you don't sleep so well tonight. Antibiotics are not a highly researched area in the pharmaceutical industry. There's a few reasons for that. One is, if you think about it, the antibiotics, a good antibiotic will take out the bacteria in a week, 2 weeks. Okay? If you have high blood pressure, you could be on that medication the rest of your life. All right? So for a pharmaceutical company, it's a really poor investment to invest in antibiotics. Not only is it a short-term treatment, but also bacteria like to develop resistance to these antibiotics. So you could spend billions of dollars developing an antibiotic, and what eventually happens is bacteria develop resistance to that, and then now all that billions of dollars you spent is wasted. So it's a really poor investment for a drug company to research these. So there are very few antibiotics being developed by drug companies for this problem of antimicrobial resistance. So this is just my first plug for academic research. A lot of this research is being done in academic laboratories where the profits aren't the main interest, it's learning the science and developing new technologies in laboratories. Another example of using these natural molecules is in Taxol. Taxol was discovered in the bark of the Pacific yew tree. So a fungus that grew in the bark of Pacific yew tree produced Taxol, and it discovered to have anti-cancer activity, anti-tumor activity in a lot of different cancers. So great, we've got a molecule that could cure different kinds of cancers. That's great, right? Well, there's only tiny amounts of it in the bark of this Pacific yew tree. So what do you do when a company says, we want to do phase 1 clinical trials, phase 2 clinical trials, phase 3 clinical trials with Taxol? So you start calculating how many Pacific yew trees do we have to grow and destroy to get enough Taxol to treat these. And the number for the phase 1 trials came out to be hundreds of thousands of Pacific yew trees. So just an unreasonable number. Well, this is where the synthetic chemists step in and say, okay, here's a chemical structure. I can make that in the laboratory. Just give me 5, 10 years and a couple $1 million, and my research group can take care of that. And so that's what happens. Several groups have reported now syntheses of Taxol, which has alleviated some of the strain on trying to get this molecule from the bark of Pacific yew trees.
Excuse me.
Yeah.
When you do produce that synthetic molecule, then do you sell it to these pharmaceutical companies because you've done all the work, and then does the university make a little?
Yep. It's actually, the question is if you make these in an academic lab, what's the next step in getting them to, and right now there's a big development because funding is so poor for academic research, and because these companies don't have the money to invest in research, there's a lot of partnerships happening between academic labs and corporations for the academic labs to develop these, and it's cheaper for the drug companies to develop in academic labs. It's a great experience for graduate students. It's a good funding source for the principal investigator. So it's kind of a win-win situation. So it's kind of a new direction, though. It hasn't been happening that much, but it's picking up a lot as funding gets worse and worse and for basic research. So this is a Taxol-related molecule, and the reason I picked this out is because what this group is doing is they're taking the structure of Taxol and they're changing it. And this is what I was talking about, tinkering with molecules, and you don't have to worry about the structure again. But I pulled this straight out of the paper, this graphic, and you can see they have this labeled as important, this is crucial, and then this is some effect. So what does that mean? What it means is when they change these structures, what they do is they test the biological activity. So they could say, okay, we're going to change these functional groups here. We're going to change this specific part of the molecule and see what the biological activity is. And what they found when they did that is if they changed this part of the molecule, it became totally inactive in the body. Okay, so they labeled this area as crucial. Okay, you have to have this exact structure in this area for this molecule to work. So then they move on to the next part of the structure. Over here, they labeled this as some effect. So they tried a bunch of different groups there, and what they found is you could put several different types of structure there, and the molecule still had biological activity. So you label this some effect. And so any drug that's being researched, this is what they do. They take different parts of the molecule and they change it a little bit and test for biological activity, and they find out which parts are really important and which parts aren't as important, which parts can we explore a little bit more. So that's my background on how we use natural molecules. So we can use the molecules directly like salicylic acid, except we don't do that very often. We can tinker with the molecules, which is done a lot, finding molecules and changing the structure a little bit. Or we can replicate natural molecules, which is a big investment too. This fourth category is kind of something totally different. Because the synthesis of natural products is ultimately feasible. Like I said, we have we're synthetic organic chemists. We have a lot of trainings in making new molecules. So if you give us a structure, we say we can make it. Like I said, it might take 5, 10 years and millions of dollars, but we can make this molecule. All right. But sometimes there's a process in nature, like photosynthesis, that we try to replicate, and we can't just take the molecules that exist in nature and replicate those exactly, because it's too complex. The process is too complex. Like for instance, in like photosynthesis, the body can utilize dozens or even hundreds of molecules to work together to accomplish a specific process. And that would be almost impossible to replicate in a non-biological setting, in a test tube if you want. So what we do is we use nature as our inspiration, and we try to learn from nature what nature does, but we try to make it better. We try to make it simpler so that we can actually do it in a laboratory. So one of the reasons we want to replicate photosynthesis is because of this. It's really just solar energy. And when I say solar energy, there's solar thermal, but what I'm really referring to is solar electric, where we can take the sun's energy and convert it into electrical energy. Now what photosynthesis does is it takes the sun's energy and it makes it into chemical energy. So it takes the sun's energy and it uses that energy to convert carbon dioxide and water into carbohydrates. And then we eat those carbohydrates, we use that energy, and then we emit carbon dioxide and water, and the cycle is repeated. And so that's how we get our energy, that's how the plants get their energy. So this process is nice, it's something we like to repeat. In order to do this, what we have to do is understand solar energy. And I got a little carried away with PowerPoint graphics here. But anyway, so usually when we think of solar energy, we think of the energy from the sun. So we think of sunburn, and we think of the energy for plants to grow, and we think of how hot our cars get in the summer on the inside. And that's all correct to make that association, but it really doesn't tell us anything specifically about what that energy is. When the energy comes from the sun, what is that energy? The answer is it's made of a bunch of waves. And I don't know if you can see this prism. You've probably all seen prisms before. If you shoot white light into a prism, it breaks it up into a rainbow. Everybody's seen a rainbow before. And the reason it does that is this white light is actually made up of a bunch of different types of waves of energy. And when those different wavelengths hit the prism, they come off at different angles. So when I say a wavelength, each one of these colors has a different wave associated with it, and the wavelength is just the energy between there. The longer the wavelength, the lower energy the light. So in this rainbow down here, the red energy is the low energy light, the violet's the high energy light, everything in between has intermediate. So every color has a different energy associated with it. And the reason that's true is because they have these different wavelengths. Now, to understand how this light interacts with matter, how it interacts with photosynthetic molecules, how it interacts with any molecules, we have to understand a little bit about the atom. And I'm oversimplifying the atom. I'm oversimplifying a lot of things tonight. So here's just a basic planetary description of the atom. So we have the nucleus, we have an electron that rotates around it. Now this electron goes around the nucleus at a specific distance. It doesn't change so much. And we can think of it as distance, it's really an energy, but that's okay. Now we can think of there being like a magnetic attraction between the nucleus and this electron. There's an attraction that holds those. Okay, so what does it take to pull that apart? If you have two magnets and they're close together and they're nearby, you have to put energy in to pull those apart. And that's what happens when a molecule or an atom absorbs light. When light comes in, that light represents energy. When that energy hits that electron, it can bump that electron to the next thing. So it would be like energy hitting Earth and us bumping up to Mars's orbit, okay? But not going in between, okay? The idea is the amount of energy that it takes to go from here to here is a specific amount of energy that corresponds to a specific wavelength of light. So maybe it's yellow light that makes this electron go from here to here, or it's red light. But it's not white light, it's not all wavelengths. In chemistry, we refer to this as quantized energy, meaning it has a certain value and it's not greater than or less than that value. So when this happens, when a molecule absorbs light, the electron gets promoted to an outer shell. You pull it away from that center, and now we have this excited state. Now the things about excited state